Good evening to those of you who are joining this session. My name is David Sexton. I'm the head of genome technology and informatics at Biogen. I am joined today by John Ellithorpe, who is the chief product officer at DNAnexus, and Frank Notaft, who is the technical director of healthcare and life sciences at Databricks. So the agenda for today is I will be speaking about what the UK Biobank actually is and why Biogen was not prepared for data at this scale. John will be speaking about large-scale analysis of the UK Biobank data using DNAnexus and how DNAnexus helped us in scaling out the analysis of this data. And Frank Notaft will be speaking about combining the best-in-breed cloud architectures to accelerate UK Biobank analysis, and he will also speak to how Databricks has helped Biogen to scale out a solution. So what is the UK Biobank and how has Biogen used it to discover new therapeutics? So the UK Biobank is the premier data set for associating disease to genetics in the world right now. So this is a long-term study of genetic predisposition and environmental exposure to the development of disease. This data set has been collected in participants who are aged 40 to 69. This is over 30 years of phenotypic data, and these patients are being followed over the long term, measuring their health over that 30-year time span. There are 22 centers across the United Kingdom with over 500,000 volunteers, and this is one of the largest and most detailed population studies ever undertaken. And you can see that there are a large number of papers that have come out of the UK Biobank. So what genetic data is collected by the UK Biobank? In 2017, 8 partner companies came together to form the Life Sciences Genetics Consortium. As part of this consortium, we will be sequencing the exomes of 500,000 of the UK Biobank participants. An exome is the protein coding region of a gene, and as part of the consortium, Biogen will be probing all of that genomic region for the protein coding genes in the 500,000 participants. The participants of the UK Biobank Consortium, Regeneron and GSK, sequenced the first 50,000 samples, and all 500,000 participants are to be sequenced in 2020. Industry partners will have exclusivity until 2021, and we currently have 300,000 exomes in-house. So how is Biogen leveraging the UK Biobank data? We are using human genetic evidence to rank our drug portfolio. We are using the data to find new gene targets, and we're using it to understand neurological disease biology. So Biogen had some informatics challenges in using this data. So our data infrastructure had challenges in not having enough storage capacity in the data center. So the UK Biobank data will be approximately 1 petabyte of data, and we did not have that storage currently in our data center. We had issues with our network and not having enough bandwidth to transfer all of this data across to our data center. And when I came into Biogen in 2018, we had just had a 1-week outage of our high performance compute cluster. So we really needed a new data paradigm for Biogen. And this is where DNAnexus and Databricks has helped us. So we needed to scale our infrastructure to deal with petabyte data set sizes. We needed to store and visualize our genomic data. We needed to analyze this data at scale, and we were required by Biogen IT to be cloud first in storing and using our data. And so now I will turn it over to John to talk about scaling the UK Biobank data using DNAnexus's Titan and Apollo products.
Thank you, David. So when we look at the UK Biobank and what Biogen needed to do, it's useful to look at the steps that are required in processing a large genomic data set. And so we really break this up into two different pieces. So there's one piece which is an upfront piece where we look at uh, preparing the high quality data set. So the data that's coming off a sequencing machine are these raw reads, and you don't uh, get the understanding of exactly what the variants are. And so there's a processing step that needs to happen across the entire 500,000 exome data set to be able to build that um, high quality genomic data set. And there's a second piece of combining that with the health and assessment data that's like demographic data and other types of data that get brought in as well, um, that then needs to get combined into a large corpus of data that then scientists can actually go and use by querying the data, asking questions of the data, and do statistical analyses to get to the final result of the data. When we look at the UK Biobank data, that data set is challenging because of the sheer size and complexity of the data. So if we look on the right, you see on the genomic side, we have 500,000 participants in that data set. Each participant can have up to millions of variants that they're tracking across these that now give you essentially trillions of data points that are needed to be able to understand what that looks like purely on the genomic side. On the so-called phenotypic side, which might be demographic information, health information, clinical information that might come from medical records, that is a wide data set. There are a lot of different fields. There are over 3,500 different phenotypic fields that are there, and they're also quite complex in that you might have significant encodings of the values. You might have hierarchies in terms of what the values could be. You also have some level of longitudinal data because people came in multiple times into the assessment centers to measure things like blood pressure and things like that. So there's a longitudinal aspect to the data. And so once you combine all these together, you have essentially a very large data set that you have to manage to be able to do the things that David mentioned uh, needing to do. So what the when we look, let's focus now at the different stages of this process, and we can talk a little bit about how DNAnexus help. In this first stage, with 500,000 samples, you end up with about 2 million files as you have the alignment files as well as the output, so-called VCF files, as well as index files that go along with it. We computed that it's around 1.5 petabytes of data that you have to process. Um, and what's really important about this data is you really need to do it in a high quality, consistent manner. And so if we look at the stages within that, you might take the raw reads and then you do a so-called alignment step, which is aligning it to a reference genome to really identify where the different fragments stack up, and then identify for every point along the exome, like what exactly is the call at that point in that SNP, along with a variety of quality control steps and such. And so uh, uh, this data set was processed at the Regeneron Genomics Center. They got the samples and were processed. They have a cost optimized pipeline that takes about 4 hours to run per sample. And so across the 500,000 samples, we're looking at millions of hours on machines. And so that's a large scale problem. Uh, and so if we look at the technology that they use the Titan product to be able to process those. And I think that when we look at why this is hard, if you're processing a few samples, it's actually not that hard to do yourself in the cloud. But once you get into the levels of thousands, tens of thousands, hundreds of thousands of samples, you really want to do it consistently and efficiently, then the fault tolerance into the clouds are very important. The ability to focus only on exceptions and the science and not having to deal with cloud optimizations and such is also quite important. It's moving data around and making sure that you have consistency in the data integrity is also incredibly important. And for a research environment, it's also quite important that you actually use the tools that are particularly important and relevant to you. And so these are all the pieces of what Titan can do. And for example, we had another pharmaceutical company as part of the consortia, and they reran 100,000 samples, processing about 1,000 exomes an hour. And so, and in 3.5 days, we're able to process 100,000 exomes. And so, like that capability is really required at scale as you go and process or potentially reprocess the data in a secure compliant environment. If I move to the next stage of the process, now we have to combine that large corpus of genomic data with this clinical data. And while there were 3,500 or more fields, it ended up being something like 11,000 plus columns that needed to get stored in the system, and then combined to be able to effectively query the data. As part of the consortia, we provided a cohort browser that allowed the researchers to visually go through and interact with the data and query and ask questions of the data, explore the data. I'll show that in a second. And what was quite important about that is that the amount of data, so the data is both wide and deep. So we ended up with something like 50 billion rows to go across the entire genomic data set down to the variant level, as well as needing hundreds of tables to manage the columns of data. And so those made real-time interaction with the data quite difficult. And so what we did is that this is built using our Apollo technology, which takes the high quality data sets that are being processed out of Titan and combines it with structured data that is the health assessment data, and provides you an ability to interact with it in a variety of ways. It's important that with a data set this large, you can't move the data around to the researchers and to the tools. You really need to bring the tools and the researchers to the data. And so this is the core aspect, and the core engine that's powering Apollo is a Spark-based engine. And so that Spark-based engine is one that is using Spark SQL to be able to query down into that data. But in order to do this, we needed to do a few things. So one is we needed to be able to partition the data heavily based on genomic location to really be able to query this quickly, as well as do vertical table splitting, essentially vertical partitioning, to be able to query fast enough because there's so much metadata across 11,000 columns. You have to be very intelligent how to actually query to be able to get response time in the seconds across this data set, and be able to really explore visually rapidly to be able to get that. And then as well as being able to provide data sciences the capability through Jupyter notebooks and other mechanisms to be able to enter the data and do statistical analyses and such with what the tools and the Python libraries or R scripts and things like that they'd want to do. Another important aspect is to keep this all secured in our environment, is that we've integrated the Spark Hive Metastore into our platform access control model to really control the security of the massive data set. And so that was incredibly important as we build this entire system together and provided it out to the authorized researchers that were part of the UK Biobank Consortia.
Excellent. Well, thank you very much, John and David, for leading into this section. I'm Frank Notaft. I'm the technical director for healthcare and life sciences at Databricks. So I manage our worldwide technical technical efforts, be it on the product development side with our genomics runtime, be it in our solution architecture team that works closely with customers, as well as in some of our functions working with partners like DNAnexus. And what I'll talk about is, how the Databricks platform comes into this, and then how we've worked collaboratively between the DNAnexus, Biogen, and Databricks teams to achieve success in analyzing this large-scale UK Biobank data. So this slide summarizes what our Databricks platform looks like in the genomic space. The way that we, if you're familiar with the Databricks platform, we have a kind of a cloud infrastructure layer that optimizes the machines you work with. And then we have a top layer that provides notebook functionality that makes it easy to use notebooks in a reproducible and shareable way. But in the middle layer, we provide a number of different runtimes that provide optimized software stacks for various different tasks that a customer is working on, be it something like processing large-scale streaming data sets, doing machine learning, or as we introduced in 2018 and made generally available last year, we've introduced a runtime specifically targeted at genomic data workflows. Our workflow covers the whole gamut of tasks, from initial data processing through the large-scale statistical analysis of variation data. We've been able to use all of these workflows at Biogen, but to focus on a couple of different points. On the upfront execution side, what we've gone ahead and we've done is we've taken the GATK best practice pipeline. So for those of you who are familiar with genomics, GATK is a standard pipeline, standard set of pipelines for taking single individuals, raw DNA sequencing reads and turning it into either germline variants or mutation calls if you're looking at cancer data. We've taken these pipelines, we've made them easy to use through a one-click interface that will set them up and run them. It takes about 5 minutes to get the pipeline set up, making it a lot easier to access these. We've also extensively performance optimized them and made them work well with Spark. So ultimately, we've been able to do things like reduce the latency of running the GATK germline pipeline on a high coverage whole genome from 30 hours to under 40 minutes, by having about a 2x performance improvement from a CPU efficiency perspective, and then using the power of Spark to parallelize this work across many cores. We've then gone ahead and we've taken a very big focus at working on population scale data. We actually have extended support for the GATK's joint genotyping pipeline. So this is the pipelines that takes data from many single samples and blends it together into a single population. We've accelerated that and parallelized that out using Spark. And then we've worked to package up a couple of open source libraries, HAIL, which comes from the Broad Institute, and GLOW, which is a project we've actually developed here at Databricks in conjunction with the Regeneron Genetics Center, that allow people to go ahead and merge these data sets together, quality control them, and ultimately run large-scale statistical analyses on top of that data. Our ultimate ambition here is to move people to an architecture where they're able to use open source technologies like GLOW that make it easy to use many different languages, be they Python, R, SQL, on top of genomic data, coupled along with efficient, optimized open source file formats like our Delta Lake file format, which we open sourced, so that they can go ahead and accelerate the process of taking large genomic data sets, wrangling and cleaning them up, joining them with a variety of different data types, be they clinical data, be they images, be they other lab measures, and ultimately produce a set of GWAS results or other statistical results that they can do machine learning on top of to generate scores, and that they can go ahead and serve directly to research and clinical audiences. When we look at some of the work that we've done on top of the UK Biobank data at Biogen, I'll go ahead and highlight some of the work that we've collaborated on with David's team around some of their genome-wide association pipelines. So with the GWAS pipelines, what this is essentially a statistical kernel that takes every single genomic variant in the data set and the phenotypes that we're interested in, and goes ahead and performs some sort of a statistical test to see if these two are associated. This could be, if I'm looking at, let's say, a common continuously distributed variable like height, this could be something like a linear association between every single genomic variant and every single phenotype. Or if I'm looking at something more complex, I might be using, more complex tests like a Cox proportional hazard model or something like that. With the UK Biobank dataset, this is particularly challenging because the amount of data that we're dealing with is very large. UK Biobank has over 2,000 phenotypes, as well as over, with the exome sequencing data, there are tens of millions of variants that are associated. So when you go ahead and do that full cross, you can be looking at running billions of regression tests to associate this data together. With the pipeline that we were able to build out, first we were able to go ahead and use the open source Hail tool to ingest this data very rapidly and start generating our first results. When these results were generated, the Biogen team was able to take some of their traditional annotation pipelines. These are tools that take those variants that we found that seem to have some sort of an association with the disease that we're interested in, and add additional functional consequences to them. Is this a variant that truncates a protein? Is this a variant we've seen in other diseases? Is this a variant that we know changes how a gene is expressed? They were able to take a pipeline that previously took 2 weeks to process 700,000 variants and accelerate it tremendously. They were able to annotate 2 million variants then in 15 minutes. So they had orders of magnitude acceleration there. And ultimately, this gave them a rapidly queryable database of genotype-phenotype associations with joined-in consequences that allowed them to really understand how these variants function and what these variants did. Ultimately, and this was really exciting. Just earlier this month, the Biogen team released a preprint on some of this work that summarizes the effects that they found in variants that are protein-truncating variants. So these are a genetic change that causes a gene to be truncated. So you don't get the full copy of the protein. You instead get a scrambled copy that doesn't produce the correct thing. They've been able to find a number of variants in about 6 different genes that have a significant impact on human lifespan, and they've been able to understand the biology of complex diseases a bit better through that. Ultimately, when we look at how all of this pairs together, the great thing that the Biogen team has been able to achieve is they've been able to achieve an architecture where they're able to use their own cloud environment, data coming from the Regeneron Genetics Center from this UK Biobank cohort, and they've been able to blend the DNAnexus and Databricks platforms together. Ultimately, this gives them a best of breed solution where they have access to the DNAnexus platform with many best practice pipelines and best practice visualization tools across both the Apollo and the Titan project. So they're able to quickly spin that up. They're able to quickly run the pipelines that they need, ultimately generate the visualizations that they need for their bench science and clinical teams. And they've been able to use the Databricks platform to do a really deep dive into these workflows. So ultimately, by going ahead and combining the capabilities of these 3 teams together, Biogen's expertise in the data, their expertise in the science, the great tooling that's available on both the DNAnexus and Databricks platforms, we've been able to take a large set of challenges. Taking this massive amount of raw data, over a petabyte of data from 500,000 individuals, complexities around a traditional ecosystem that Biogen had, and a need to move to the cloud. And ultimately, the Biogen team has been able to deliver a lot of success with the findings that they have from this very large scale database of comprehensive variation linked to comprehensive phenotypes. They've been able to go ahead and identify new drug targets. They've also been able to build out models that allow them to understand how genomic variants impact the functionality and the possible success of other drugs that they've been developing. So they've been able to go ahead and reposition and reprioritize their drug portfolio. And ultimately, as you look at the complex neurodegenerative diseases that Biogen is working on, data sets like this give them a lot more insight, a lot more precision, a lot more ability to interrogate the complex biology of neurodegenerative disease. So as every month passes, as we mine this data set more, we are all developing as a community a better understanding of complex human disease with the power of this combined genotype and phenotype data. And ultimately, I've been very excited, to see the collaboration between the DNAnexus and the Databricks teams. We see a tremendous amount of overlap where customers can benefit from both of these technologies, be it how they're using the Titan project for many of their data processing needs, Databricks for much of their ML, and they're leveraging a lot of the technologies for visualizing, understanding, and interrogating genomic and phenotype data that are available in the Apollo project. we see a tight integration coming up as there, Spark ultimately underpowers many of the different technologies in this space, and we're looking forward to collaborating a lot more. We're very interested in talking to to anyone out in the field who is interested in using these products together and who would like to influence our future roadmap on how these products integrate together. Thank you again very much, David and John, for for joining us on this adventure. I think everyone in the genomics community has been really thrilled to see how the UK Biobank data is giving us a ton of just tremendous insight into the complex biology of human disease, and I really greatly appreciate your partnership in these efforts.